Okay, so we'll now continue with this idea of extents. This, again, is a fairly basic, straightforward concept. We had this, we found the center of mass and center of volumes before, and that gives us some idea of where the object is. But if we want to know something about how big the object is, then we can start using tools like extents to get an idea for this. So rather than just having the area, what are the actual dimensions of this object? Another name for this is bounding box, and so some tools will call this a bounding box, others will call it something else, or bounding cube if you're dealing with 3D images. And that basically the idea is to find sort of the minimum and maximum points in every direction. This is sort of important to note because if you do have a single voxel that's turned on in sort of a far corner of your image, that will be part of your bounding box. So it won't take any sort of reasonable analysis and say, okay, this is probably too far away to be part of it, or this is not close enough to the center or something. This takes the entire range. So if we now look at this, we end up with an object, like this car before, or from this car image, where we want to be able to define the extents of it, and we do it in a very basic way, like we've done all of these things, where you kind of loop through the image and find all the coordinates and add them on. All of these are entirely illustrative. So if you're ever doing a project and you're writing code like this, you've probably done something horribly wrong because you shouldn't have to write these loops. These loops are just here to make you aware of kind of how these things work internally. They shouldn't actually be part of any image analysis you do because all of these functions have been implemented very carefully and efficiently in other libraries by other people. And so these are just really to show you this is how, kind of the pseudocode of how it's actually being done, but this is an incredibly inefficient way to do it. If we were to look, finding the extents takes, 1/10 of a millisecond. There shouldn't be any reason to go through each point manually, which can take very, very long. We then take sort of the minimum and the maximum, so we can then print out that sort of the variation in x is from 4 to 12, and the variation in y is from 3 to 15. We can then do sort of a visual representation of this, where we then draw this as a bounding box. So we sort of show this is the bounding box we have for our object. And that's quite easy to represent once we have the original image. But as I said before, if we have this image like here, and we go seg-image, so we turn that 1 pixel on. We can then rerun this, and we see that the x range goes from 0 to 12, and the y range goes from 0 to 15. And now if we redraw this bounding box, we end up with a bounding box like that. And so if your segmentation isn't good, or your segmentation has problems or has errant pixels that show up, your extents and bounding boxes will be sort of equally bad. And so it's important to make sure all the segmentation and all the sort of cleanup operations has been done before you start with this task, because if there's a problem with that, it will sort of make these results very difficult to interpret. So we can go back because we don't actually want to save that. And show what it should look like normally. We can then go to sort of a more concrete example where instead of just having one car, we take the original scene we were looking at before with the component labeling done. So we've now labeled all of these cars as separate objects. We can now start to do the shape analysis. So we've come up with a few very basic metrics for what we're doing, where we can run through all of the pixels. Yeah, so here we use the region props function, because again, we don't want to be going through all the pixels manually, and that then does the shape analysis for us. And then all we have to do here is extract the right parameters, so the centroid, the bounding box, and then draw them on top of the image. And so we end up here with the 9 regions, and we have a point at the center for each car, and a bounding box that surrounds it. And so this, is already a fairly useful representation, that by looking at this, you can kind of see what's coming into your algorithm, what it's identified as boxes. So for kind of starting to debug these things, it's easier to use this than it is potentially with that. And then what we're really interested in is extracting out single values. And so this is where we go through all the components we have and we make a table, so that rather than having images with thousands of voxels, we have a very simple table. And so you can look at the code here if you're interested. The most complicated part of this is how you figure out the name of a color from the RGB values, but that's not at all related to this course. And so here what we have is we have sort of for each label a row where we can show the area of that object, the bounding box, the centroid, the color, and then the label. So the label doesn't really mean anything, but we can get out sort of these sub-regions and this sort of analysis. And so now we can look at this table and very quickly say, how many powder blue cars did we have, or how many silver or gray, or any number of other sort of questions that we might want to ask about the cars that we have. We can also start to begin to filter to say where, probably things with 25 pixels are not really cars, and so we'll leave them out, or, less than 30 or 40, because even looking at them, it's very difficult to see how that might be a car or not. I mean, I think even if you gave this to a human, they would be completely overwhelmed with the task of trying to identify what that small pattern of pixels were. Whereas with the other ones, they seem to be fairly reasonable. And so, making decisions like that, of course, is much easier once your data is in sort of a tabular format and you can start to analyze it. So now in terms of other metrics, so if we look at this table, the most interesting metric we had was maybe color. The bounding box and centroid give us something about the shapes and dimensions. Maybe if we had, if we knew how far away it was, we could calculate the actual size of the car. But, if we want to start doing more interesting analysis, we'll want to quantify these shapes a little bit better. And so anisotropy is a useful metric for a lot of the analyses we'll do. For cars, it's potentially less interesting. Although if you were trying to distinguish between a car and a truck, you might say cars are kind of more square-like, the same size in most directions, whereas trucks are much, much longer in one direction than the other. And so if you were to use a metric like anisotropy, maybe that would help you distinguish them. But for cells, it's often quite important because, healthy cells are fairly round, and cells that are dividing can get elongated before they divide. Cells that are sensing certain forces often have a shape that reflects what kind of forces they're sensing, and a whole number of other issues that you can use to start to characterize these things. And so anisotropy is a very sort of vaguely defined term. Varying in magnitude according to the direction of measurement is the official definition. And correspondingly, there are hundreds of different metrics for how you calculate anisotropy. And so when you read scientific papers or look at works people do, everyone picks whatever value of anisotropy or whatever formula they like the best. But there's not any clearly defined metric which is the standard definition for anisotropy. So you'll need to be aware of that when something mentions anisotropy, there's not an obvious thing that you can use to recalculate it unless they give you the formula they did. But typically, the values are such that if it's a sphere or a circle, it should be 0. And the bigger the value is, the more anisotropic it is. But as we see here, we'll do a quick example with sort of synthetic objects, and that's what's always very helpful for these kind of analyses is when you make images yourself. And so here we make sort of an xy plane, and then we make an ellipsoid function. So basically, the x squared over a, or x over a squared plus y over b squared is less than 1. And so what this allows us to do is sort of generate whatever shapes we want to, so that we can calculate the anisotropy for them. So here we have the anisotropy functions, which are the 4 functions I mentioned above, implemented as sort of code. And so now what we can do is we can take a shape. We start off with a perfect circle. We show all the different ones on the axes, and we can sort of show how those values change as we change the shape. And so the first thing we do is we make it longer, and then we make it skinnier. And so we'll see that, the first one kind of, when we have a range from 0 to 3, goes off to almost infinity by the last point. The second one is sort of much better behaved, goes from 0, gets a bit bigger than 1, I guess. And the last two, the third one maxes out at 1, and the fourth one maybe goes to 2, if I'm not mistaken. But fundamentally, they all behave slightly differently. And so when you're trying to calculate statistics on objects, it's very important that you know which metric is being used. And particularly, or what do you think would be the biggest problem if you were looking at something like average anisotropy with using one of these formulas? So if you have 1,000 cells and you want to say the average anisotropy is X, is there certain ones you would use over other ones? Or do they all seem about the same and you can just randomly pick? So either looking at the distribution or looking at the formulas and sort of thinking about it. Yeah?
Some metrics have a much higher dynamic range, I guess, so
yeah.
Higher variation.
Right. So if we look at like metric 1, yeah, it has a huge dynamic range. And so you could have one object that was very goofy shaped that then has a value of 995,000 on the anisotropy scale. And your average anisotropy for 5 cells, even though only one of them was oddly shaped, becomes 200,000. Versus if you have a value that goes between 0 and 1, and you have a bunch of spheres and one really oddly shaped object, the mean value is much more likely to be in sort of that range. And so when you're using metrics that go all the way to infinity, so you're dividing by something that can get down to almost 0, you need to be incredibly careful when you average them, because one huge outlier can affect that distribution a lot. And if you're using an anisotropy between 0 and 1, outliers won't really affect you. Because, when it goes between this shape and this shape, the change here, it's almost a perfectly linear curve. And if you're dealing with something where, having incredibly anisotropic objects makes a massive difference, so maybe it's, energy conductance, and a really anisotropic object can conduct much more electricity or much more efficiently, then it might make sense to use a metric that gives you that larger dynamic range. But for most things, having that large dynamic range means that if you do something like averaging or any statistical analysis afterwards, it will be quite difficult to interpret what that value actually means. So in terms of principal component analysis, who's heard of it before? Everyone. Does anyone feel comfortable enough to define it? Sure? Yeah.
It's a way of finding the direction
variation inside of a set of data.
Yeah.
However you define variation and however you want to define direction, but usually just a vector representation that gives you the vector along the greatest variation of data. And if you take n of them, then you get the n greatest directions, all orthogonal.
Yeah. Good. That's correct definition. And what's it useful for?
A lot of stuff.
Any specific examples?
Dimensionality reduction.
Yeah. And so what does that mean?
Basically, if you have like 1,000 features, like you want to like get the most 500 most important features in the data set.
Yeah. Yeah. No, I mean, that's perfectly right. I'm impressed. No, I think last year there were very few students who had heard of principal component, or maybe they were just very sleepy. But yeah, so it's a very important tool. You come across it in a number of different scenarios. In finance, it's used for, predicting, for figuring out correlations between, the price of corn and the price of gasoline. I find one of the examples that resonates the most with me, and hopefully a bit with you because you're scientists as well, or many of you are, is sort of spectral analysis. Because this is an example where you have a huge number of variables, and it's often very difficult to kind of do other approaches with them, and so PCA tends to work very well. So is everyone familiar with infrared spectroscopy? Or Raman spectroscopy, or visual light, or some kind of spectroscopy? Has anyone never done spectroscopy before? Okay. So we'll cover it a little bit, but basically the idea for spectroscopy is that you have a number of different wavelengths. So you send white light through your sample, and you measure the intensity at different wavelengths of the light coming out. And you see fundamentally what's in your sample, or what your sample made of, by what it absorbs or what it interacts with. And so this can be very useful in areas like biology, where we have sort of a simple, somewhat synthetic example here of, you have, here's the wave number. And so that's sort of what we'll use for wavelength here, and then you have the intensity. And so you have this range between 1,000 and 4,000, and you're going through a biological sample. If you have nothing, you kind of have this noise background. If you have fat, you have a peak here and a peak there. And if you have protein, you have a peak here and a peak there, and maybe a smaller peak here. So these curves aren't completely accurate. They're simulated, but it gives you sort of an idea of what your data look like. And this was a problem I was faced with when I was an undergrad at some point. And the solution we decided to take for this was to find the peaks. So, you write a little code that goes along each point and tries to figure out if it's higher than the last point, or if it's higher than the average. And then you find sort of the peak. And then maybe you fit a Gaussian curve to the peak because you know something about the distribution. And so to take one of these data sets, find all the peaks, and spit out all the results, can take, first of all, quite a bit of time. It's very difficult when there's so much noise in the data. You see lots of little variations here that make it quite difficult to find the peaks. And, this peak and this peak are not very different anyways. They're very similar to one another. And so it's, finding exactly the center and exactly the width is not always easy. And you have to have some idea of how many peaks you're looking for, because if you look for 50 peaks, you'll probably find 50 peaks, but not all of them will be sort of significant. Is that clear? And so basically what principal component analysis allows us to do is to be incredibly lazy. And so we just take all the data that we have and throw it into an algorithm and have it figure out for us what's important. And so what we want to do is really, how can we analyze, separate these groups, figure out what the major differences are in these groups in sort of a quick, efficient way. And so like you mentioned before, you can use PCA to take these thousands of dimensions and reduce them and find a bunch of orthogonal representations that are useful for your data set. And so here is kind of the data set we have. We basically make 20 curves, 60 curves of protein, fat, and noise. And we sort of add it all together and we have this big data set. And so we show the first 4 curves here where we can see, you know, curve 1, curve 2, curve 3, curve 4. The noise background is a little bit different in each one. And that's sort of the result that we have. And if we were to try to group the points together by just taking, you know, the first and the second point on the axes, we would end up with this sort of complete mess of points there. And so what we want to do is we want to find the dimensions or find the points that explain the most variance in our data. So how can we find a representation of this spectroscopy that explains the data well? And so that's where PCA comes in. So again, it's, you know, you should never have to implement PCA yourself. It's in every toolbox you've ever used. I think probably Excel even has a PCA feature in it somewhere. So, you know, with what we're doing, it's in scikit-learn decomposition. You give it PCA, you tell it how many components you want to find. So here you can make your life easier by saying 5 instead of 1,000. And then you give it the test data. And so when you run this, this is done in seconds. I mean, it can run through huge amounts of data very quickly because it's been implemented efficiently. It's a tool that lots of people use. And what we can see is that, here, where we can now show the different principal components. So we can show, you know, the first component that it found is this red curve. So we sort of have a little bit of this peak, a little bit of the second peak, and then a lot of the third peak. And then we see that the second component is sort of a bigger peak here, a bigger peak there, and sort of a negative peak there. And so the components themselves we're not incredibly interested in, but it gives us an idea of sort of what's happened in our system. And that when we take each point that we put in and we plot the value along component 1 and the value along component 2, we see that it's done a very good job of separating our data sets. And so what we have now is we have sort of, these clear separation between sort of what is the noise and what is the signal. That's what the first component's really focused on. Is there anything present in the sample at all? And the second component is then really emphasizing what's the difference between fat and protein. And so that we take an incredibly noisy data set, we put it in this tool, and it's come up with a way of automatically classifying these things into different groups and compressing our data set down into just 2 dimensions. So that rather than having intensities at a number of different wavelengths, we can just keep track of component 1 and component 2. And we're able to classify any new sample that comes in as being noise, protein, or fat. We're also able to take samples that might be somewhere between protein and fat and quantify sort of where on this axis they fall. And so it gives us a number of very helpful tools for analyzing a really complicated data set like this without having to manually identify each peak or try to fit curves or doing a huge amount of noise removal, because noise sort of by default is just this large variation. And so that will be gotten rid of when you look at what explains the most variation in your data. We can also then show sort of how much of our results are being explained with the components. And so what we see here is that sort of the first and second component explain almost everything, and that the other components explain very, very little. And so for us, that's also quite comforting that we know in the first 2 components, we have over 50% of the variation explained, and that probably the rest of it is just noise if these components are so small. And so it makes our task quite easy to simplify the data and just worry about these first two components where we can come up with simple rules, or even if we start to use approaches like we covered last time with sort of decision trees, it's much easier if the components you're putting in are understandable. So if you just say the 0 is the signal curve and 1 is the protein curve, then you can have decisions based on that rather than decisions based on thousands of different wavelengths and noise patterns that might be coming in. If you go through the scikit-learn documentation, they have a nice example with this for face analysis. And so they have a data set that's made up of all thousands of different faces, where, yeah, you can use this sort of scikit-learn data sets to fetch this Olivetti faces data, where it's sort of randomly shuffled. There's 64 by 64 images. They're all centered in the middle, and so it makes it very easy to look at them separately. We can then show the images themselves, and so we see the face of all the people. We can then use Pca to make a decomposition to figure out what the eigenfaces are. So that's very similar to what we just did with the spectra, where we found the eigenspectra that sort of explain the differences, where the eigenfaces sort of explain the differences in the face data set. So we see, where this image has a high intensity are things that were very different between the faces. And so here we see that for the second component, eyebrows were quite important. Here, beard or non-bearded seemed to be a fairly important one. I guess that one could be glasses as well. And that you can start to understand, most of these faces can be made up from adding a number of eigenfaces together with different weights and coefficients. And so this gives you sort of a way for taking all of these images and decomposing what are the largest sources of variation or what are the biggest differences in your samples, without having to assume anything or build any models. Of course, if your faces aren't all centered, this becomes very problematic because then you'll just find out that the position in the image is sort of changing a lot and you won't get useful components. But if the images are all centered and noses are all in the same positions and eyes are all in the same positions, then you can start to see kind of what features differentiate faces from one another. And sort of go back to this representation where you're able to say, this face is equal 0.5 of this plus 0.7 of that plus 0.8 of that. And you can add, beards to people who don't have beards and other sorts of analyses like this. And so it gives you a useful decomposition of the kind of data that you're looking at. So that was all a very, very, very long intro to what we're actually going to use it for, which is a much simpler sort of analyses. And so what we will take Pca and use it for is helping us sort of find the orientation of 2D and 3D objects. And so, when we looked at the bounding box, this wasn't very helpful because it simply finds the largest box around your sample. And if we use shape analysis or Pca analysis, we can find kind of the optimum orientation by finding the principal components of all the pixels in our image. And so the way we do this, the formula for principal component analysis, you've probably all seen before. We kind of do this covariance first, and then you do this eigenvalue decomposition where you come up with these eigenvectors. And so that was the eigenspectra in the cases of the spectroscopy, or the eigenfaces in the case of the faces. And for this is then the primary orientation vectors that we have for the image. The eigenvalues then represent the strength along that axis. And so this we'll find makes up a useful representation for how big the image is in that direction. And so rather than using sort of the caliper length of the bounding box, we can take the eigenvalue to tell us sort of how long or large an object is in a certain direction. And so we can take this image we had again, where we have all of these pixels that belong to the image. and we can perform a PCA analysis. And so basically what we see here is that we can create a PCA, we put in all of the XY coordinates, and we get out a rotation of all the pixels in the image so that the greatest variance is along the 0th direction. And so here is the greatest variance, here is the second greatest variance. And so it kind of gives you that optimal rotation so that your sample is longest in that direction. We can then plot that on sort of the original data where we have sort of the first component and the second component of the image, and how that now gives us a way of describing how the sample is oriented. That we can look at this and see, okay, this is the primary vector. We can calculate the angle between that and the x-axis, or that and the y-axis, or that and other objects, and begin to analyze the sample in more detail. And this is a nice approach because it works equally well on 2D and 3D data. So it doesn't require any assumptions about your axes. You simply put the data in and you get the result. And it's very quick. So even if you have thousands and thousands of points inside your object, it works reliably. And because, as we showed up here, you have this sort of covariance representation, it's much less sensitive to single points than the bounding box approach was before. So we can actually go here and we can do the same thing we did before where we say scikit-image, where we turn that voxel on, or that pixel on. And we can now do that variation, and we see that that pixel is still there, but that it, well, it flipped the direction. But other than that, it didn't really change these components or their lengths very much. So that you see this doesn't go all the way down to that pixel anymore. It goes down a little bit further, but it's not completely skewed by one single value being wrong. And so when you're dealing with data sets that have lots of noise, you can deal with that noise much better if you're able to sort of use distributions rather than just maximum and minimum values themselves. So we can go back and get rid of that point. And the directions that come out are not meaningful. So cells don't have orientations, or these objects don't have orientations. There's no way to know the difference between this part and this part. And so this arrow is just shown as a representative arrow. It could just as easily be in the opposite direction. And so when you're looking at these, you shouldn't be too, you can't make any meaning of the direction itself. You can only make meaning of sort of along what vector is it going. But that the flip of that would be just as acceptable and has nothing to do with how the data is distributed. It has to do with sort of the initialization of the principal component analysis. So sort of the take-home message is we can calculate distributions for these x, y, and z coordinates, find kind of the correlations, and rotate it. So it gives us a very reliable way of finding the orientation of the sample and extracting that in a reliable way so that we don't have a bounding box or something that's very sensitive to single pixels. It also gives us a nice representation of what we're looking at. So this can be looked at as sort of an ellipsoidal representation, so where you draw an ellipse around the object and you use the sort of the square root of the eigenvalues as the length of the semi-axis. And so you can then make an ellipse from any collection of pixels that you have by performing this principal component analysis and then drawing the ellipse with these appropriate values. So I think we'll stop there if there's no questions, because that's sort of the last part that's relevant for the lecture that's coming up next week, the week after next. So I think you have a week off for Easter, and then April 12th will be the next lecture. Are there any questions over those topics? So the exercises this week, I think, have been updated at this point. One of the more useful ones, I think, that you're now slowly getting enough tools to be able to do well is this MNIST shape analysis. So this one, this EM cell segmentation is also a quite useful one. This is a data set that's from EPFL, and so it's electron microscopy data of 3D segmentations, and so it's how you can segment these data. I think these are also used in the KNIME workflow, and so if you're interested, you can use it there. But you have these sort of large TIFF images with objects inside, and it's how can you go about automatically segmenting these objects. And so if you look through the notebook, you have sort of these grayscale images here, and the goal is to kind of automatically find all of the little structures inside of it. And so in this example, you'll just go through and you'll be given the structures by some other segmentation, so a manual person who went through and identified them. And you'll do some kind of shape analysis to group them into different categories. But of course, it's also interesting to try to figure out how you can build an algorithm that can segment those fairly reliably. And then, yeah, you can create the bounding boxes. You can see the different shapes. You can find the axes that explain the most difference in the samples. You can calculate anisotropy. And so it's very helpful to actually apply these to real data sets and get a feeling for how that works. And then I would say the more pragmatic example to do is where you have this contest. So this MNIST digits recognizer contest. So it's on Kaggle. You can download the data set yourself if you want to, and it's about recognizing these different digits. And so what we do in this example is to take all of the images that come in, perform a segmentation and a labeling, and then a shape analysis. And so we'll take, the anisotropy, the area, the perimeter, and try to classify the digits. So we build a classifier to automatically take the digits in and guess which one it is based on a shape analysis. And so what you'll see is you can build a classifier fairly easily. You put this value in, it guessed 9, the actual result was 5. So it didn't do a fantastic job. It was also not particularly confident. So when you look at the other things it was evaluating, it didn't do a great job. You can then prepare your output. And so this has everything you need to make a submission to the contest, where you can then go to output, submit to competition, and see where you actually rank with a classifier you've built yourself and a task of classifying, 60,000 digits. And so we see our score is 0.26. And so that puts us at place 1986 out of, does it say somewhere? 2034, I guess. So using all of the approaches from this class, you get a pretty poor rank. And if you look, and this is even more depressing, where is it? So of course, all the ones that say beginners that get 99% accuracy are also a little bit, yeah, see KNN from scratch with Python. And you can see that they were, did he submit it? Oh, we can't submit that one. But for all the ones that did submit, I think you see next to their name what they did. And just using a simple simple approaches get you 0.99, so get you in the top ranking. And so the challenge becomes, how can you improve the shape analysis or add new features or use things like principal component analysis to actually make something that's better at classifying digits. And so, yeah, as I was mentioning earlier, I think this is a very useful way to test out approaches and ideas because it gives you immediate feedback. The data sets are very small, so it doesn't take you a month to process them. You know, you can process 60,000 images in a few minutes at the most. And you can see how you perform on a leaderboard that you had nothing to do to set up. And so here, you know, we could obviously find lots of ways to improve the notebook that we had. We used, you know, anisotropy. Clearly, anisotropy alone wasn't a very good feature. Maybe, you know, the orientation would be a better feature. And so we can use the Pca analysis to find the orientation and include that. Or we could try to, you know, break up the object and analyze the separate pieces of the object. Or you could try to take sort of the a thinner version of the object, because maybe that's easier to process and you have less noisy data, and see how well that works. And so there's lots of different approaches you could try to make a better classifier, but you really have the advantage of being given immediate feedback of if your features and if your model worked well. Because when you do these things yourself, you get very, you know, maybe you get feedback when you submit a publication or when you show it to your advisor, but you get very minimal feedback, and very few people are actually able to tell you in an absolute manner how well your analysis worked. And so with this, you're able to get immediate feedback and process a lot of different things very quickly. And there's, of course, more complicated data sets. So there's data sets where you're supposed to classify, I think, CFAR 10. It's another very popular one, where it's about recognizing, I think, 10 different categories of objects. And so maybe a little more relevant for some of the image processing cases that you normally deal with. But still very small images that you can process very quickly. And so here you have cars, birds, dogs, boats, trucks, and your goal is to classify the different ones. And so this one, if MNIST isn't interesting or is exciting enough, you can try these data sets. And it's a slightly more complicated problem, and so maybe has better use for some of the features you come up with. There's also a data set called Fashion MNIST, where they have 10 different types of black and white objects from fashion, so shoes and bags and pants and shirts, and you try to classify them into those 10 different groups. And so there's lots of little practice data sets where you can run very complicated analyses in a number of minutes on thousands of different examples to actually see which ones work well. So yeah, so I'd recommend kind of getting a feeling for these shape analysis tools and then trying one of these contests to see if you can apply the tools and figure out which features work well or what features you might need, because it'll give you a much better feeling for how these things can be applied to real problems. And of course, what the talk in 2 weeks will be about is how you can take thousands of images like this from data sets that come from these automated robotic microscopes and automatically decide if, you know, a cell is alive or not, if a drug worked or didn't work. And so you have these sort of binary classification decisions that you're trying to make with them as well. And so these examples are much more similar to the kind of work that Roche and Novartis do with drug discovery, because they do take a lot of images as input and they need to make decisions, and they need the decisions to be reliable. And so you want it to work for thousands of different images with different kinds of illuminations, with different scales. And so doing that well requires features that are very robust. And playing with these data sets will kind of give you the impression what features are robust and what features are incredibly sensitive to noise, and so they don't really help you classify very well. So if there's no questions, we'll stop there, and you can start working on the exercises or enjoy your holidays.